Key reading. The title of the article is Microadaptations. Microadaptations means small adaptations. Microscope research. Introduction. We are Bárbara, Linda, and Miguel Ángel from APAMA. We are doing a project for the journal Agua Simple. We are going to study biodiversity, which means different living things. We will study biodiversity with materials that we will explain later. For a few weeks, we have learned to use the materials to research water samples. Objectives: to learn how to use the magnifying glass to observe different living things. To learn how to use a microscope to see small animals. Learn to use the book called The Ultimate Visual Guide to the Natural World. Materials. The materials we have used for this project are as follows. We use the magnifying glass to observe the animals. With the magnifying glass, we made the animals bigger to see them better. The book called The Ultimate Visual Guide to the Natural World. We used the book to look up the common and scientific names of each animal. The microscope adapted to the tablet. We used the microscope to take pictures of the animals. The slide to put on top of the animals. Then we put the slide to the microphone. We used the cover slip to see the animals better. Methods of research. We learned how to use microscopes. We saw objects under the microscope. A leaf, a finger, a sponge, a dry leaf. We took 3 water samples from different areas of Madrid. The first sample was from the pond in the Sierra de Guadarrama. The second sample was from the Jarama river. The river was near the factories. The third sample was from the pond in the APAMA orchard. We researched these samples through the following steps. We learned to use the book to do exercises about animals. We learned to focus with the microscope and magnifying glass. We put the slide on the table. Then we take a drop of water from the sample with the dropper. We put the drop on top of the slide. Then we take the cover slip and put it on top of the drop. Now you're ready to see the animal under the microscope. Next, we focus the microscope with the wheel to see the animals at different sizes and distances. We look through the microscope for an animal. Then we take the tablet and take a picture of the animal under the microscope. We search in the book or the internet for the common and scientific name of the animal. Finally, we write the name of the animal. Research results. We saw different animals in each sample. We saw these animals in the pond in the Sierra de Guadarrama. They are the animals that live in clean water. Ciliates verticella, daphnia magna, copepoda, notonecta. In the Jarama river, we saw these animals. These are animals that live in dirty water. Remains of living organisms, and daphnia. Pinolaria, daphnia magna, torrent planaria. In the garden of APAMA, we saw these animals. These are animals that live in fountains and parks. Microscopes albifrons, Apis fabae. Acknowledgements. Linda, Bárbara, and Miguel Ángel would like to thank the APAMA Association and Victor Pascual Ayuso. They have helped us complete this project for Agua Simple.